Okay, so time to get into some detail about this cell cycle, which is basically just the cell's lifespan. What are the activities that it goes through basically from its life, through its life, from its creation to its death, basically. Concept 12.2 says the mitotic phase, which is the part that we're talking about, where they divide and make more of themselves. The mitotic phase alternates with something called interphase in the cell cycle. So cells have lots of jobs to do. Dividing and making more of themselves is not the only thing that they do. If you're a muscle cell, you've got to move the body around. If you are a cell as part of the digestive tract, you have got to take in nutrients. So all the cells have an actual job. Cell division and reproduction is just part of what they do as far as their life cycle. So a lot of times the cells are not in a dividing state. They're in something called interphase. And in 1882, there was a German anatomist, someone who studies the body and the various body parts. His name was Walther Fleming, developed dyes to observe chromosomes during mitosis and cytokinesis. So this was helpful in being able to see and view what was going on inside these cells. Still a long time ago, way back in the 1800s, so the ability to apply dyes to the cells that he was looking at was helpful to kind of illuminate what was going on inside. And so what he noticed was that there was a great period of time where the cells were not actively going through division, were not actually reproducing. And this is what we now call interphase. The period between where the cell divides, it does grow, and it appears to grow larger. It makes more cytoplasm and things like that. But there's more to it than just that. There's a lot of important events that occur in this interphase where the cell is growing and doing its job, basically doing what it does in, instead of reproducing or before it begins to reproduce. So we're going to divide the cell's life into this cell cycle into a couple of different parts. One of them is the mitotic phase. That's the actual reproductive part of the cell where it's making another copy of itself. And that's mitosis, where it duplicates the DNA, and then cytokinesis, where it divides itself in half. And then the other part we're going to look at is called interphase. And interphase is basically everything else. That is the period of time where the cell is growing and doing its normal job. It's also copying materials and organelles, and then begins the copy of the chromosomes in preparation for cell division. So most of the time is spent in this interphase, where it is either growing or doing its job or preparing to divide, and then you have a smaller phase called the mitotic phase, which is where the actual division occurs. So this interphase makes up oftentimes a huge portion of the cell cycle, about 90% of the cell cycle, and we can kind of divide it into three subphases. The first one is called the G1 phase, and you'll find out the G stands for gap, and we'll find out why in just a moment. So the G1 phase is a first gap. Then we have the S phase, synthesis, and then the G2 phase, the second gap. And the cell grows during all of these phases, but the chromosomes, the DNA, is duplicated in that S phase, which is why it's called synthesis. Of these three phases, the S phase oftentimes takes the longest period of time. So of these phases, which make up a huge portion of the cell cycle, the S subphase generally takes up a bit more time than the G1 and G2. So here's a great picture taken from your textbook. This I really like because it kind of helped me understand and put a drawing or an illustration to the words. So here we have the entire cell cycle. If you close the loop, you have the entire cell cycle. Notice that the vast majority of this cycle is in something called interphase. And that's the portion where the cell is growing and doing its normal everyday work, and also preparing for this reproduction mitotic phase. So right here, just this 10% of the time, it's in the mitotic phase, and that's mitosis, which is dividing up that DNA that was replicated, and then the cytokinesis. So of course, at the end of this, you have two cells, two daughter cells, and then each one of these cells can go through this entire cell cycle again. Of the interphase, you've got it divided into G1, S, and G2. It depends on the cell. Oftentimes the S phase, where the DNA is being synthesized, tends to be the largest of the phases, takes up most time. In this model, they've pretty much just divided it equally, but it actually depends on the cell. So interphase is made up of G1, S, and G2, and then the mitotic phase is the actual part where it's dividing into two. Now, for much of the chapter, we're going to actually zoom in on this little area right here. So even though the cell spends most of its time in interphase, we're going to zoom in on this little area called the mitotic phase, and we're going to subdivide that into some further defined parts that you've actually probably heard of before. So that mitotic phase, mitosis, is divided into 5 subsections or 5 phases, and you've probably heard these before. Prophase, when I was in school, we didn't really use prometaphase. So when I was in school, it was just prophase, metaphase, anaphase, telophase. Now they've kind of subdivided and added another phase there. So it's prophase, prometaphase, metaphase, anaphase, and telophase. And then we have cytokinesis, which is the actual dividing of the cell material in the cytoplasm. That kind of happens in the last 2 phases here. It kind of overlaps and starts to occur somewhere around anaphase and finishes by the end of telophase there. So cytokinesis kind of occurs the same time as the last 2 parts of mitosis does. There's a great Bioflix movie on mitosis, so make sure that you go into the files for Chapter 12 and find the Bioflix movie on mitosis. Click on that and hit play. I don't think it'll play in the PowerPoint here, but go and find that movie separately and take a look. Again, your textbook does a fantastic job of illustrating certain concepts. So I would turn to page 232 and 233, and it's laid out very nicely. This basically all of the steps of mitosis, starting with G2 of interphase, which is the last stage of interphase, and then going through prophase, prometaphase, metaphase, anaphase, and telophase, and then cytokinesis, sort of finishing it off. So we'll take a look, but also I would highly suggest going to your textbook, because the textbook not only shows the pictures, but it gives further information below each one of these phases. I don't expect you to have every single bit of information that the textbook lists memorized, but you do need to know the basics of what's going on in any particular phase. So if we look at the very end of interphase, we have the DNA that has already been replicated. Remember, that happened in the S phase. We have the nuclear membrane, nuclear envelope is still fully intact, surrounding the nucleus, surrounding the DNA. This is the plasma membrane on the outside. We have centrosomes, which were also reproduced, replicated. So there's 2 centrosomes, and they have 2 little centrioles each right there. Then we're going to move into prophase. In prophase, the main thing that starts happening is you start to get a condensing of the chromosomes. So the chromosomes begin to basically fold on to themselves, and then they start to become visible, and they condense, kind of start to form that X shape of the 2 sister chromatids that we have seen. And we start to see what's called the mitotic spindle, which we'll talk about in just a moment, where we have microtubules that begin to emanate from the particular areas here, the centrosomes. And this is going to build the framework that's going to stretch the cell apart and also allow the chromosomes to line up in the center. Then we have what's called prometaphase. In prometaphase, basically the nuclear envelope has self-destructed or dissolved, and so the nucleus is basically open and free. We have the microtubule structure pretty much complete from pole to pole or from end to end, and we start to see the chromosomes begin the process of being attached to these microtubules and beginning to line up in the center of the cell. In metaphase, through an action that we're going to discuss with the microtubules kind of pulling back and forth, we have a lining up of the chromosomes pretty much down the middle of the cell, sort of on an invisible line here that we call the metaphase plate. And we have the centrosomes at the different poles. They have this framework of microtubules running from one pole to the other pole. In anaphase, those sister chromatids pull apart and began to move down toward each pole. And then finally in telophase, we have the sister chromatids that are now separated chromosomes. They begin to unwind, and the nuclear envelope begins to reform around them. And then cytokinesis begins to pinch off the 2 cells until they become 2 whole separate cells. Along with illustrations, your book has some great microscope pictures here using fluorescent stains to kind of highlight certain amounts of material, which is really cool. So in G2 of interphase, the bluish that you see is the DNA. Not very clear because remember it's still very thin strands at this point. It already has been replicated. We can start to see some of these microtubule strands that are going to make up those filaments. Then we have prophase. You can see the genetic material is beginning to thicken or condense. We have prometaphase here. And again, it's quite clear now. And now you can see the centromere area here. In metaphase, all of the chromosomes, the sister chromatids, have lined up in the center of the cell. In anaphase, they begin to move, be pulled apart, and then are moved toward the poles. And then in telophase and cytokinesis, you can see that that division is almost complete, and they're pulling apart and separated into two separate cells. Great, beautiful microscopy here. So take a look in your pictures inside your textbook, again on page 232 and 233, and then also read below because there's some great information below. So we'll look at each one of these sort of individually here. So this is part of interphase, the last part of interphase called G2. The chromosomes have been duplicated, and the centrosomes have also been duplicated. That's going to be where the mitotic spindle emanates from. The chromosomes not yet condensed, so it's kind of difficult to tell one from the other. They're just kind of a big mass in there. Prophase, the chromosomes have begun to coil and thicken, so that makes them easier to see. The sister chromatids start to become visible. The mitotic spindle starts to form. Prometaphase, the nuclear envelope, the nuclear membrane has basically fragmented, and the microtubules, you can see those tiny little yellow strands of microtubules begin to extend and make this framework. Metaphase, the centrosomes are at each kind of pole or end of the cell. The chromosomes begin to line up along the center line. They're pulled back and forth, we're going to find out, by the microtubules that line up on this imaginary line called the metaphase plate. In anaphase, which tends to be the shortest of all of these phases, by the way, the sister chromatids are pulled apart and they split, and the microtubules pull them down toward the poles, or sometimes actually there are motor proteins that walk the chromatids down toward the poles. And then in telophase, we have basically two nuclei, and we have the nuclear envelope beginning to reform. The chromosomes are beginning to unravel, and then cytokinesis is beginning to squeeze that cell into two. Let's take a closer look at a major portion of this, which is called the mitotic spindle. The mitotic spindle is that framework of microtubules that you saw. They are responsible for kind of lining out these chromosomes, getting them all centered, and then pulling them apart and moving them down toward the poles. So the mitotic spindle is a structure made up of microtubules, which we've discussed in previous chapters, and they control the chromosome movement during this whole division process. In animal cells, remember, part of the animal cell is called the centrosome. It's the microtubule factory or organization center. It's usually located very close to the nucleus. As part of interphase, when the DNA is replicated, centrosomes are also replicated, and those two centrosomes will move toward the two opposite poles of the cell to kind of start this whole structural process for mitosis. The movement of those centrosomes toward the ends happens pretty soon, pretty early in the process during prophase and prometaphase. Within those centrosomes, there's two little parts, a pair of what we call centrioles. Those centrioles also play a part in building this mitotic spindle and organizing this, but there have been some experiments where it shows that the centrioles don't have to actually be there. They've done some experiments where they have removed the centrioles from a living cell, and still it can build the spindle fibers that it needs to build. And in plant cells don't even have centrioles, and they still build this spindle fibers, and they still build this framework. So it's not really totally understood what the purpose of the centrioles is. They do seem to help the process, but the cell can actually do without it, which is kind of odd. In a lot of microscope pictures, you see this cool, almost like a starburst effect coming from the centrosomes. So in here, we have all the chromosomes have lined up at the middle along that center line here in metaphase. And then we have the two centrosomes that are kind of at the opposite poles. And look at this starburst of microtubules that's coming out. That starburst of microtubules is called the aster. It's a radial array of these short microtubules. So we call this entire thing the spindle. And that's the centrosome and all of the little microtubules and this aster that you see here. So there's an aster at both ends of the cell. And all of this microtubule framework is called the spindle, or the mitotic spindle. Now I said that these microtubules kind of attach to the sister chromatids and line them up along that center plate during metaphase. Well, what they do is they attach to something called kinetochores. Kinetochores are proteins that are going to be at the same spot as the centromeres at that waist area of the sister chromatids. Remember, the centromere is that little narrow waist of the little sister chromatids that kind of form the X, kind of in the very center there, where the two sister chromatids are most closely attached. Well, there at the centromere, there is a protein complex called the kinetochores, and the kinetochore is the anchor point. That's where the tubes, the microtubules, actually attach to either side of these sister chromatids, right there at what's called the kinetochores. So they are the anchoring point for these microtubules. So in prometaphase, what begins to occur is these microtubules begin to build this framework, and they connect to the kinetochores, and they begin to pull back and forth. So you have microtubules pulling from both sides of the cell, and they're basically kind of in a tug of war, pulling these sister chromatids back and forth and back and forth. And that process begins to align the sister chromatids at the center of the cell along that imaginary line that we call the metaphase plate. So with enough time, this kind of tug of war back and forth, this attachment to the kinetochores pulls the chromosomes kind of into a center alignment to where they're all lined up in the center of the cell, ready to be divided. This is another great illustration out of your textbook here. So it shows some of the different parts in a dividing cell. This would be about in metaphase, when they're all kind of lined up in the center. So you have the aster. There's that kind of starburst of microtubules. There's an aster and an aster down there. You can see the centrioles down there as well. You can see the sister chromatids are sort of lined up in the center. Some of the microtubules are attached right there in the center, attached to the kinetochores. So if you zoom in, like on this one right here, right there is kinetochores attached to that waist that we talked about, that thinner portion right there at the sister chromatids. And then here you also see some of these microtubules are not attached. So not every single one of these tubes is attached to a chromosome. Some of them go from one end and attach to others at the other pole, and they're actually going to be used to push this cell apart. They're going to overlap right there. They overlap, and they're going to ratchet away from each other and kind of extend this cell out and elongate this cell. So some of these microtubules help elongate the cell and push it apart, and some of them are attached to the sister chromatids right there at the kinetochore, and they're responsible for kind of pushing and pulling these chromosomes until they line up right there at that center imaginary line. Finally, when all these sister chromatids are lined up in the center, they're going to be separated. They're going to be pulled apart during anaphase. So in anaphase, the sister chromatids separate from each other, and they began to move down the kinetochore microtubules toward the opposite ends of the cell. And it was not really understood exactly how this worked for a long time. One thought was that the microtubules basically shorten at the poles, and it's kind of like reeling in a fish. If you are reeling in a fish, you are shortening the line at the end where it connects to the fishing rod. And it was kind of thought, well, perhaps that's the way it does it, that the microtubule is basically reeling in the chromosome toward the poles. And then another thought was maybe there's motor proteins that are walking along the microtubule, and they're carrying the chromosome down toward the pole that way. So it was actually best understood now as a combination of both of those. There's been various experiments that have shown in different varieties of cells that both of these processes are actually going on. So in many cases, the chromosomes are walked down the microtubule by a specific motor protein, and in some cases they are sort of reeled in by the microtubule from the pole. The microtubules shorten by depolymerizing at the kinetochore ends. So for the most part, what happens, and I'll show you a picture so it makes it clear, is the chromosome is walked down the microtubule, and as the chromosome passes an area of the microtubule basically is being disintegrated, depolymerized behind it. So imagine you are crossing a bridge, and as you walk, the planks of that bridge fall off behind you. That's basically kind of what we're talking about. And a picture always helps, so let's take a look at an illustration. So one of the ways that they discovered this was in a particular cell, they had the ability to stain or mark the microtubules. So here we have the sister chromatids that have pulled apart and they're moving toward the opposite poles. Here's your aster at the various poles. And what they did was they marked a particular area on the microtubules. And then they watched the process. And if you look closely, you'll see which side of the mark is shorter. Well, you'll notice that it's the side that's closest to the kinetochore, the side that is attached to the chromosome. So this area is still the same length here and here. But if you look carefully, this is quite a bit shorter than it was up here. So what's happening is this is kind of zoomed in. This particular chromosome is being walked along by a motor protein. This is kind of a track that it's following down toward the pole. And as it passes that area of the microtubule behind it, the microtubule is basically being taken apart. And here are the tubulin subunits that made up that microtubule. So again, imagine you're crossing a bridge, you're walking over across a gorge, and the bridge is being taken apart behind you. So every step you take, the planks behind you are removed. It's kind of the process of what we're talking about here. Now, I did mention that not all of these microtubules are attached to the chromosomes. Not all of them push and pull the chromosomes and move them toward the poles. Some of them are actually a structural type of microtubule that overlaps with the opposite one from the opposite pole, and they actually help push against each other and elongate the cell. So if you take a look at the microtubules on slide number 37, let's back up just a little bit. So right here, see some of these microtubules, they're not attached to the chromosomes, but if you look closely, they do overlap each other. And where they overlap, they have the ability to push off of each other and stretch this cell, elongate this cell. Let's go back to where we were. So in telophase, we have genetically identical daughter nuclei form at the opposite ends of the cell, and then you have the cell being pushed apart by these microtubules, and then basically those chromosomes begin to unravel, uncondense, I guess you could say, and the nuclear envelope begins to reform around it. So you kind of have a proper nucleus at the end of telophase. The very end of this would be the finishing part of cytokinesis, where the cell pinches in half and the cytoplasm is divided and the cell membrane pinches in half until it becomes two separate daughter cells, and they are duplicates of each other. That last stage, cytokinesis, kind of an interesting way that this happens. In animal cells, it's a little bit different than plant cells, but in animal cells, remember, we don't have a cell wall. All we have is a kind of a soft, squishy cell membrane. So in animal cells, there becomes a small indentation or a small furrow called a cleavage furrow right in the middle there, where it's basically being pinched down. So that forms what we call a cleavage furrow, and that forms a groove basically in the center along that imaginary line called the metaphase plate. And then what happens is there's a ring of actin microfilaments, and you may have remembered we talked about actin and myosin, which is responsible for muscle contraction. So there's a ring of actin microfilaments that surrounds this groove, and it acts with the protein myosin to begin to pinch together. It's almost like a drawstring. So imagine you took a balloon and you tied a string around the middle, and you had it like in a lasso to where you could tighten the string, kind of like you're pulling the drawstring on your shorts, or you're pulling the drawstring on your laundry bag. And as you pull that drawstring, you pinch the center down and it becomes smaller and smaller. That's what this ring of actin microfilaments is doing with this myosin protein, pinching the middle until it becomes two separate cells. Now in plant cells, it has to work a little bit differently, of course, because we have a plant cell wall. So in plant cells, what happens is they form what's called a cell plate. there is no pinching down in the middle. It forms a temporary sort of building wall. So we don't have a cleavage furrow. What actually happens is the Golgi apparatus creates vesicles that contain cell wall material, and those vesicles move toward the center of this dividing line, and they begin to join together. So these vesicles that contain cell wall material, which would be cellulose type material, they join up together to form something called a cell plate. And then they begin to basically build outward until they reach the perimeter of the cell and fuse with the existing cell wall that's already there. Here's a great little animation. You have to right-click and hit play. So here's an animation of cytokinesis in animal cells. I think it also shows the plant cells as well. So again, right-click and hit play. We have two animations here. This one is kind of a live shot taken through a microscope of a living cell going through mitosis. That's kind of cool to look at, and there's some good text alongside with that. Again, right-click and hit play, or it may start to play automatically if you're looking at the PowerPoint. And then this one is, again, a sea urchin that's going through mitosis, the cells of a sea urchin going through mitosis, kind of a time-lapse photography taken through a microscope. So right-click and hit play. Or if you can't see this and you want to see this, you can't make it work for you, just let me know and I can post this online as well. Some great pictures in your textbook side by side showing cytokinesis in animal cells and plant cells. Here in the animal cell, these are SEM shots, scanning electron microscopes. You can see the cleavage furrow where it kind of looks like there's a string that is being tightened around the center of the cell. And you can see in a drawing illustration that cleavage furrow that will eventually pinch the cell into two. That's an awesome shot taken through a scanning electron microscope. And then over here, this is a transmission electron microscope. You can see this cell wall being built. So you can see it's made up of little blobs or vesicles that contain cell wall material, and they are building that new cell wall in between the cell plate. Let's take a look. I think we have some closer up shots. So again, here's the animal cell going through cytokinesis, the cleavage of the animal cell. And here's a closer shot of the plant cell showing those vesicles that came from the Golgi apparatus forming that cell plate, and eventually that will join together and join up with the existing cell wall. Some great shots of the entire process in plant cells. Sometimes plant cells are easier to see. You can tell it's plant cells because you see these nice cell walls that look nice and brick-like. So here's prophase, and then you have prometaphase where the chromosomes are beginning to condense. You have metaphase where they're beginning to line up in the center. And obviously at this point the nucleus has basically, the nuclear envelope has disappeared. Here you have anaphase where they begin to pull apart toward the opposite poles. And then you have telophase where you can see they become two separate cells and that cell plate is beginning to be built. Some closer views here. Prophase, prometaphase, metaphase, anaphase, and telophase. Another way that I used to mention this or talk to my students about this was sort of like thinking about it as a dance. It's kind of funny to think about these chromosomes kind of lining up like a square dance to where they line up in the center and then they separate down to the poles. So I've seen entire little video clips that kind of compare this to a chromosome square dance. And it talks about the movement of the chromosomes during all of these stages. So prophase, prometaphase, metaphase, anaphase, telophase. Again, in your textbook, great shots, great pictures that show you exactly what it looks like. All right, we're going to finish off this portion by talking about a slight variation on mitosis. What about in prokaryotic creatures like bacteria? So we spend most of our time talking about eukaryotes because humans are eukaryotic, so that kind of makes sense. But there's a process in prokaryotes, which would be bacteria and the archaea. They reproduce by a type of cell division that's slightly different from mitosis. It's called binary fission. Binary means two, fission would be to divide, so that makes sense. It doesn't work exactly like mitosis, but it's kind of their version of reproduction / mitosis. Some of the differences is that there's no visible mitotic spindle. You don't have this framework of microtubules. You don't even see microtubules, as a matter of fact. And it works a little bit differently because they don't have a nucleus. They don't have everything that the eukaryotic creature does. In binary fission, you're going to have the chromosomes replicate. You have to have a duplicate copy of the DNA, of course. And we'll show a picture or a drawing in just a moment. But this starts at a spot on the chromosome or a spot on the DNA molecule called the origin of replication. And it's just a particular spot that's kind of like the starting point for this division or replication. And then as it divides and moves down the line, it begins to separate. And one strand of DNA kind of becomes two copies, two separated chromosomes, two daughter chromosomes that actively move apart. They're not attached together like the two sister chromatids or anything like that. We're basically sort of taking a single strand of DNA and creating two copies. Almost in a way, it kind of looks like unzipping a zipper, to where a zipper has an origin of replication, a starting point, and then as you unzip it, it's like the one part becomes two. The only difference is we're making a copy. So it's not like a zipper where you're basically dividing it into half and then you have two halves of a zipper. We are duplicating the DNA as we move down and create two copies. The plasma membrane then pinches inward, and you have a kind of version of cytokinesis dividing the cell into two, but it's not exactly like mitosis. The exact nature of how all of this occurs, how the daughter chromosomes move to the opposite poles, which proteins play a part, that's still under debate and under investigation because there are no microtubule structures. So the exact process is still kind of being studied. So that's cool. I love it when we come across things in science that we're still studying, we're still learning. That's the beauty of science, is you never have all the answers. So here's a great illustration from your textbook. So this would be a single-cell bacteria, a prokaryotic. It doesn't have a nucleus or anything like you see. The DNA is just floating inside the cytoplasm. So you have this long strand of DNA. Oftentimes the bacteria only have a single long strand of DNA. And there's going to be a particular point in this big long strand called the origin of replication. And that's where it's going to start to replicate itself to make another copy. And then it's basically, you can see, sort of pulling apart, and it's going to move down the line. And as it moves down the line replicating, it will take those two copies and move them apart. So we'll kind of add to this. That point of origin, see where it's kind of subdivided right there and pulling apart? Well, those 2 points of origin will move toward the opposite poles, and this replication process will continue. Eventually, you'll have two distinct copies, and you'll get kind of this cytokinesis going on, sort of in the center. And you'll end up with two daughter cells, two exact duplicate copies of themselves. So this is binary fission, how it occurs in a prokaryotic creature, say like a bacteria cell. Lastly, for this particular concept, talk a little bit about the evolution of mitosis. Since prokaryotes were around long before eukaryotic creatures, since they evolved millions and millions of years before eukaryotes, mitosis probably evolved from binary fission because that's the process that prokaryotes use. And that does kind of make sense when you look at some of the information and you look at some of the evidence. Binary fission and mitosis does share a lot of characteristics. A lot of the proteins involved are very similar, and they have some of the same characteristics. So that kind of helps make it clear that they're probably related in some way, or perhaps that one evolved from the other, that mitosis evolved from binary fission. There's even certain living creatures that kind of follow a process that falls in between. So there are certain protists, which are the small unicellular creatures of a wide variety. They exhibit a type of cell division that almost looks midway between binary fission and mitosis. And two great examples are dinoflagellates, diatoms, and some yeasts. I guess that would actually be three examples. So on page 237 of your textbook, you can see a great illustration, and I'll show it in just a moment, of these three different creatures that kind of do a variation that falls in between what a bacteria cell does and what a human cell does. Dinoflagellates are unicellular protists. Diatoms are a type of unicellular algae, and yeast, you're probably familiar with that, that's unicellular fungi. So all of these are eukaryotic, like our cells are, but they're much more, I guess you could call them much more primitive. And the way they divide is perhaps midway between binary fission and mitosis. So here are the two, or all the examples kind of lined out. So at the top we have a bacteria cell, and it goes through binary fission. And then we have these guys, the dinoflagellates, some diatoms, and some types of yeast. And if you watch what they do in their reproduction process, it's sort of midway between what our eukaryotic cells do and the bacteria cells do. So they have some of the same structures, but not all of the same structures. They have some of the same processes, but not all of the same processes. For example, if you look here, they have microtubules, but notice the nuclear envelope never disintegrates. All of the separation and replication of DNA occurs within the nuclear envelope, and that's not what happens in us. And then you come down here to the diatoms and the yeast. They also have an intact nuclear envelope, and they have the kinetochore microtubules, which these guys didn't have that. So these examples kind of fall in between where our cells and the bacteria cells do things differently. So this kind of shows you an evolutionary pattern, perhaps how this basic version of reproduction changed over time to become the version that we know in eukaryotic cells. We're going to pause right there. We'll finish up with the last couple of concepts in just a moment. Take a break, come back, and I'll see you in just a few minutes.